Next talk will be given by Marc Flesch from Genedata on an open and collaborative enterprise software platform for patient and compound profiling.
Thank you. Yes, that would be great. Hello, can you hear me? Is it working? So hello everyone. So I'm Marc Flesch from Genedata, and I'm going to give you an overview about first what our company does. So we are not EMC and we are not Bayer, so I probably should drop some words about where we are originating from and what's our company focus. And then I'm going to give you a brief overview of the collaboration that we had with Bayer over the last half year. So we have a long-standing relationship with Bayer for a couple of years already, but I'm going to focus on iRODS on this talk here. So Genedata was established in 1997 and it's a privately owned company with a headquarters in Switzerland. So we have about 200 employees as of now, and there is an office in Munich where I'm working off, and then there is 2 offices in the States, one from Boston and one in San Francisco, and there's another office in Tokyo, mainly doing support and so on. So development sites are basically Boston, Basel, and Munich. So our mission basically is to support the drug discovery and biotechnology folks, and mainly in the biotech and pharma research area. The majority of our staff is PhD level experts in computer science as well as bioinformatics mainly. So the customer base in pharma is basically all 25 of the top 25 pharma worldwide are using some of our products, and Bayer Healthcare is among those for years. But I should not waste too much time on this stuff. Let's focus on what iRODS came to us and what iRODS can give us. So to motivate it a bit is, so where we see a really good contribution of iRODS coming in the future is to support the patient and profiling process. And so what patient profiling or personalized medicine means has to do with basically sequencing patients with new techniques. And the guy from EMC was telling that there is a real drop in cost of sequencing, and this is accelerated a lot in the past with Illumina, and it's going to be further accelerated with next generation sequencing. And there is a huge value in all this sequence information for pharma companies. And these data are already there as of today. So they're already available there and are now used in patient stratification. So to try to figure out if a certain cohort of persons is a drug responder or not. And this is really one of the hot topics in pharma these days. And basically every pharma company is doing this. So what is the main issues that come up with that? And I'm just going to focus on one of these, but there is 3 things that came to our mind where we say this is the main challenges that you have when you work with patient profiling. So one of them is that the heterogeneity of the data is really, really complicated. So you have metabolic data, you have proteomics data, you have all kinds of screening data, and you have to bring all these data together to get a common understanding. So that was also in the previous presentation on the slides where you saw these studies and the project, and then below that you had all these folders, which are basically the different technologies that reflect the personal state of people. And that was all well and fine until NGS stepped in and everything got personalized all of a sudden. So, and then the field was driven by fear. So the fear was formulated in one statement from the biostatistician head at Bayer, which is we have to do something about the patient privacy. And we have to take the patient privacy serious when we're going to work with these data, because we got the data, the personalized information being handed in from our patients that we profiled, and we can't actually use them in research directly without being sure that they are treated really severely, right, and seriously with respect to privacy. So that was basically the starting point where iRODS stepped in. So the key feature that we were using from iRODS is the security feature. So the scale-out NAS and storage opportunities is, of course, also of help when it comes to collaborating between different teams. I mean, all of these pharma companies, they have sites all over the world. So if you can, in one go, also overcome the data sharing and data transportation issue and abstracting, so to say, the storage location from working with the data, that's fine. But the main reason why iRODS was introduced into that field had to do with data privacy and chain of custody, so backtracking of where the data were originating from. And I want to give you an example of what we see as of today. So when I'm going back to Boston on Thursday, I'm going to see this again. So what do we have in pharma companies these days? Looks like the Stone Ages, right? So you still have Unix file permissions, POSIX access control lists, Zamba shares, and it's really all these huge amounts of data are still under this regime. And all these companies try to fight. I mean, that all works kind of in a research environment where you don't have personalized data, where you don't have to make sure that the personalized information and the raw data themselves have to be separated. So where these two worlds, so the phenotype information and the genotype information, which is actually in the BAM files, are tied together. And so they have to somehow disentangle that in a safe manner. And at the same time, of course, you have the Illumina 10x machines coming out that get you a 30x coverage for less than $1,000 whole genome sequencing per person on a really high scale. So the volume is really going up very high. So that is, so to say, the status quo. Let me quickly say how we see or how we work with iRODS and what our product is. So we're working in the NGS field now for 5 years or so, where we started actually developing a software solution that has a data management component, a statistical analysis tool, and a data NGS processing pipeline. And so we made this in collaboration with Bayer ready for working with iRODS. And how that works is basically that we say that iRODS is a safe harbor for all this personalized information. So all the data warehousing part is in iRODS. So the input data that we are dealing with in our system is iRODS. So we directly connect to that via Jargon API. And then we transfer the data into the, I would call this the workbench of NGS, which is a GPFS or an Isilon system or whatever, with a huge compute cluster attached to that. That actually allows you to work with the NGS data at speed. So you can't have this, as Carsten pointed out, do directly in iRODS as of now. Maybe that's possible in the future if we extend the iRODS capabilities a bit more. But at the moment, there is a requirement that you have to transfer that first. Then you do your NGS pipelining, which is taken from a screenshot here in the middle. So that's what our solution looks like. And then the results are stored back. And the important bit for doing that is that you have to basically allow with direct interactions within our data processing pipeline and iRODS. So what you see here highlighted is this graph is a screenshot from our software. It represents a data flow. So we have certain data nodes where NGS tools are executed, or where certain processing steps, like for example the read mapping, are done here. And the flow is from top to bottom. And so we have in our system something like 150 of those different nodes. And roughly 80 of them we had to basically rewrite or extend to directly being able to coordinate with iRODS. So that allows us for not having any unattended data lying around on disks, checked out somewhere, where people can actually simply forget to delete them. So that it's really ensured that the personalized information is encrypted on the fly and all the time under our control and not being available to others. The reason why we had to do like this had to do with the politics within Bayer, where they said, as Carsten pointed out, that they have command line users that are early adopters that are using the system from a Unix command line directly, as well as people that are using our automated pipeline. So we have a transparent way to work with the data in both fashions, exploratory, with the people that are using the NGS latest and greatest tools that are out in the public domain, as well as with our system. So we didn't rely on our data warehousing ourselves, but with, we relied on iRODS in that, which is much easier for us to have an open standard somehow to integrate with. So this is the key value. So how is that done? It looks a bit like an electronic thing, but it's the same workflow as I showed before. So you have on the left hand side the source, and on the right hand side you have the destination, which is in both cases iRODS. So data are coming from iRODS and are landing in iRODS again. And as I said, you have, for example, in the NGS field, you have FASTQ files, as we heard before. These FASTQ files are personalized by metadata. And this metadata are then traveling through Profiler, which is our software solution, through the whole process. So these are all the AVUs that you have in the system. And then you have, of course, in the mapping step, for example, you can have multiple files that are put together into single files. So you have much more inputs that are coming here and they're coming out here. And then finally, you may have something like an expression matrix, where you have RNA expression values from all these people that you have sequenced, RNA sequencing, for example, and have all this information being tied together in a single file. So what made it was the biggest part of our implementation was to figure out what all these individual processing steps are doing and how metadata are flowing between these steps. So you have all kinds of forks and joins of metadata and agglomerations, like in this case. So all the metadata tags from all the input files are consolidated here automatically. Good. So that's the big picture. So I just have screenshots here from the software, unfortunately, and can't give you a live demo because I have the software not with me. But so we are relatively close to finishing the project. So we are currently in the sign-off phase with Bayer, within update cycles. So the end of year we should have that software running live. And it looks quite good. So I just want to show you a couple of screenshots, which is 3 things here. So one is to do with how we access the data. So we've seen a web UI before from EMC. So what we have in Profiler, it's the name of the software, by the way, is that when you select files, we have a file selector for that, which is this big UI you see on the left-hand side. And we wanted to make it as silent and as integrated as possible for the end user. So it still looks like a file selector. So the only thing we did is, so we have a path selector at the top, and you can transparently browse through iRODS collections and pass as if you would browse through a file system. And what you gain on top is that you have the additional metadata attributes being listed here on the right-hand side, and you can basically pick from these columns at the same time. So that's one thing. So that you have the raw data, which is in this case the BAM files, and the annotations being available in one file browser next to each other. And what you can also do is you can directly type, as you are used to with your Windows Explorer, search terms into the search field here, where you can, for example, search for all the BAM files that not have a control associated with this, with a certain factor level combination and a certain condition, and get them as search result. So these search results, as well as the listings in the collections directly, can then be used and selected within our system for data processing. So at any time, we are just relying on iRODS paths. So for us, it's always, that is the most important bit for us actually, because since we have these multiple sites that people are collaborating together with the same data over different sites, they can always use the same URLs. So we are storing our state of the workflow and so on in XML files. And these XML files contain, of course, the source locations from where the data were taken. And these source locations are always iRODS paths. So that's the way how we guarantee data provenance across the whole data processing pipeline step, right? And I mean, what you see here is what we also put in is a bulk editor for working out with metadata. So, I mean, in a production environment, one has to have it automated, of course, as Carsten pointed out. One has to have automatic validation and so on in place. But to make it a bit easier for the end users, at least in the exploratory phase, we allow for actually editing metadata attributes based on queries. So you have the possibility to search for something. So this gives you all the files that start with sample starting from a home folder. And they got listed here. And then you can select them all and then say edit metadata. And then you get a bulk editor that agglomerates all the attributes that have been found on all the files at the same time. And you can either filter that down, or you can have multiple attributes being listed here. For example, I've selected concentration. You can figure the system such that you be able to lock down certain attributes from editing. So that's really recommended for attributes that are required and should not be used by the end user. For example, the patient ID or the parent information, which is basically the source path where the file was coming from originally. You can also browse through the individual files of that set at the same time. So that's just the user interface that we put up so far. But the majority of the work was not in the user interface. It had mainly to do with the processing engine. So let me wrap up. So what we have learned is that if you use Genedata Profiler, what you gain from that is that you can kind of rescue how you work with the system. You can rescue the feeling of working in a research environment if you go to patient and patient profile and private data. So that is something which is otherwise really cumbersome to do, so that you have to respect the patient consent all the time. And the other thing is that with our system, you get the chain of custody for free. So we have audit trailing built in and all the other things that are required for regulatory authorities to actually validate the system. So we didn't go through the validation yet, but we are in the process of working on that, mainly at the moment. So that's my last point. So thanks for your attention. Questions?
Any questions?
Yeah, I'm very interested in your chain of custody or your data provenance, if you will. So we have a similar problem in that, data's flowing in and mixing from different files and all this kind of stuff and then coming out. My question has always been, can you go backwards through this tree? So in other words, usually instead of having to go all the way back down the branches and then back to the main branch and then all the way back out, can you kind of get back to the original from the outside? Does that I don't even know if that question makes sense, to tell you the truth.
Yeah. So the question was, just to repeat, if one can reverse the data flow and figure out from the result files where the data were coming from. And that's exactly what was one of the main requirements to do and to fulfill. And the reason for that has to do with the patient consent. So at any time it can happen that a person actually revokes this patient consent. And then you have to ensure that all the influence of the data that he provided is removed from the system. And this kind of get a tricky thing if you're thinking about a GWAS study, for example, a genome-wide association study, where you have a lot of crosstalk across patients from one person. And that is one of the main requirements that we had in the system. I didn't show that yet, but all derived data are only available until the time point that the person that has permission to read these data also has permission to access the source of the data all the time. So it's ensured that the source of the data and the permissions based on the source of the data is driving all downstream permissions. That's how we solve this. So, and we do this by tracking the patient IDs mainly. So the patient IDs are basically the main driving force for having the access permissions right. Did that answer your question?
Very good, yeah. It's, what was I going to say? Say for us, it's like an internal control. You know, we have a process building. We're doing quality control on it.
Mm-hmm.
And, you know, maybe sometimes something happens, right, in the process. Mm-hmm. And then we want to go back and see, oh, is there what's common about that happened in the past that might explain that?
Yeah.
So we might pick up things that we weren't even noticing before.
Yeah, it also has aspects of QC, of course. I mean, quality control of the input data is also one of the main tasks that is part of the workflow, so that you are not working on uncurated data, and that you have to ensure that the data quality and so on is always good. And this is information that you want to have surely also downstream when you do the statistical analysis. So the secondary analysis that you do on the original analysis has to know about the data quality as well. And that makes it also for us, we put all this information in iRODS now, right? So for us it's easy, right? We can have an experiment and simply annotate it with the coverage, which is really great because we don't have to maintain this information elsewhere. It's next to the BAM file. It's as metadata in the BAM file. There was another one.
You probably said this and I missed it, but what stage of release are you at? And if you're at beta, are you looking for beta testers?
So this is a custom development, and we did together with Bayer. And so our delivery date is 1st of October. So I hope we make that. But I mean, just to give you a bit of background, there is, from the Genedata side, it's 10 developers involved into that for 1 year now. Developing the functionality, and so we are not too bad with the work packages. So there's a couple of work packages that have been signed off and been agreed with Bayer yet, and we're going to have this as part of Genedata Profiler 9.1 being available for the public then as well.
You just showed us the ability of selecting several files and modifying all the metadata they have. Are you doing any kind of intersection between those metadata, or are you just showing all metadata each file has, or something like that?
I guess you're referring to this dialog here, right?
Yes.
So what this dialog does is actually we are taking all the samples at the same time, multi-select them, and then we have this table here on the left-hand side, and you can see that the majority of the items are grayed out. And the majority of the items that are grayed out, meaning it's kind of the same thing like iTunes does. So that was driven by how iTunes deals with metadata. So we were inspired by the music industry and MP3 tags when we designed this. So this is basically all the items that are grayed out are non-common tags on the files. And what you do is you can select one here, and then you can set a certain value on this attribute, or you can have multiple values in this attribute, and then press OK. And that would apply this to all the files at the same time. Like you do with an album artist in iTunes, right? So in iTunes, what you would do is you would select all the songs from one album, and then set the album artist in one go, right? Driven by a search. And that's how we do it. How we do that. And it's, of course, kind of dangerous because you can really screw up the whole metadata system by this. But, I mean, we put some safety features in, so that's not working with if you do that on root and press star and then remove. So you get complaints.
Maybe you mentioned it, but I think I might have missed it. Do you keep track of chain of custody for changes to metadata also, or not?
So there is two classes of metadata that are flowing through the system. So, I mean, first of all, we had to extend our system to work with units. So it's really, we had metadata in Genedata for a very long time, but we had to completely change it because we introduced units. So we had to extend that. And there is metadata that are coming into the system from the input, and there is metadata that is locked down, like, for example, the parent information. So, for example, if you start with a FASTQ file here on the left-hand side, the derived file, which may be a BAM file in between, has as a parent information the iRODS path, the full iRODS path of this FASTQ file. At the moment we are using the pass. Maybe we should use something like GUIDs instead, because that would allow for later on rearranging the file system structure within iRODS itself. But for the time being, Bayer decided on using the FASTQ pass. And we are reporting ourselves, of course, as being part of the pipeline as well. So this XML file that is describing the data processing is attached as one of the informations of the downstream processing. And we are also reporting the workflow, the way we process the data, to the result. So that allows you to actually trace back and build reports out of our software. So you don't need to have our software in place and running anytime later. Also, the biostatisticians actually can come in and check out where was this data coming from. Was that processed with Genedata software, or did we do that ourselves, or where was that coming from? And the other source of data is coming from our system ourselves, right? So QC is one of the aspects of what we do with the data. And the QC metrics that are created while during data processing are also attached to the output. And this is an automatic process. I mean, no one would go through all these, go through the files and manually put certain numbers and quality metrics to these files. So the majority of metadata that I would expect in future in NGS is fully automatic. It's nothing to do with manual intervention at all, apart from the patient ID.
You mentioned that you're using FilePass as the source and not yet using a GUID. You're dealing with heterogeneous data. Is there anything that you can use as a unifying way to extract a GUID out of the file so that if it became disassociated with iRODS, you'd be able to reconnect it later?
I'm not 100% sure if we are the source of the GUIDs at Genedata at some point, or if that's owned by our customers. So maybe they have some kind of barcoding or figuring out what the GUIDs are. But I mean, GUIDs are ideal, I would say, to work with. You can just stamp them as a GUID. I mean, you can just have a meta tag with a GUID, and you can fully search that all the time, right? So it would be very easily accessible across the whole file system later on by just doing a string search. I mean, that's how Microsoft, for example, works in the registry, where they associate GUIDs with certain tasks in the operation system to globally identify things.
So what I'm looking for is something that if someone tried to re-ingest a file, the system would recognize that it already had it based on some hashing algorithm or some other unique identifier.
I would expect that some deduplication capabilities are coming later in iRODS directly, hopefully.
That capability exists for social science data sets. They construct a unique numerical fingerprint of a file such that they can then identify that across any version of the file. We should stop here. Thank our speaker again.
Thank you.